So our last speaker here is Alexis Dodson. Motley views evolutionary impact of audience perception on morphology in the jumping spider, Synemosyna formica.
Can everybody see everything and hear me okay?
Yes.
All right, great. So thank you all for coming. As he mentioned, I'm Alexis Dodson, and today I'll be presenting some of the research from my master's thesis, which is looking at the evolutionary impact of audience location and perception on morphology in the jumping spider, Synemosyna formica. Oops, sorry. So first we'll start a little bit by talking about, we're using a Batesian mimic as our model here today, so we'll talk a little bit about Batesian mimicry. Batesian mimicry is a strategy that confers protection from predators to relatively harmless animals that resemble unpalatable or dangerous animals. So here we have two examples, a jumping spider velvet ant mimic and a caterpillar snake mimic. But if you look at these, you can see a lot of differences between the mimics and their models. And this is an ongoing quandary in the study of mimicry, is why some mimics are poor approximations to their models. If these animals are under such acute natural selection to resemble their models, why do they not become perfect mimics? So to explain this, let's start with the basic model for imperfect mimicry. Broadly, the model for imperfect mimicry suggests that there's some phenotypic attack threshold for a given predator. So if we look here at this figure, along the bottom on the x-axis you have the phenotype, and then this dashed line is the mimic's phenotype, the solid line is the model. If the mimic has a phenotype that exists in this black region, they're likely to get attacked because they don't resemble the model closely enough. And then over here in the white area, they should be safe because they look enough like their model. And this attack threshold can change based on a number of things. So there are several hypotheses for why this occurs that can be grouped into roughly three categories that explain how selection might be relaxed or how the mimic is making best compromises in conflicting selective circumstances. So the first group is the role of the model in protection. And these hypotheses look at abundance and the defense of models and the influence of those on mimetic signals. The next is developmental or genetic constraints. So this looks at the physical limitations of the mimic itself. We're going to focus and hopefully add to this last group perceptual and cognitive features of receivers. So predators may use cognitive shortcuts where they only use one or two signals to identify unpalatable prey, or mimics might interact with multiple predators with distinct perceptual capabilities. But mimics aren't only dealing with multiple predators. They also must contend with their models and conspecifics, each with distinct perceptual capabilities, all coming from multiple angles. And when we think about that a little bit more, this could be a clue for why imperfect mimicry exists. Each of these audiences are exerting different, potentially conflicting selective pressures, often from distinct locations in the space around the mimic. And mimics might adapt to each of these pressures based on the vantage points from which they come. So we're going to test this in Synemosyna formica as a mimicomorph. We're going to look at the shape from the top and side perspective and the color from the top and side perspective. So as I mentioned, Synemosyna is a mimicomorph. This is a widespread Batesian mimic strategy. It occurs in nearly every order of the insect kingdom. And it's a good strategy because ants bite, they sting, they taste bad, they're pretty small. But messing with one can often mean messing with the entire colony. So unless you're an ant specialist, they're really not worth the effort. And this is also widespread in arachnids. So we see this in a lot of arachnids. Here we have Synemosyna formica, our species of focus. When we compare this to a typical jumping spider phenotype, we see that Synemosyna has a distinct, very ant-like shape. So it has this sort of elongated and thin cephalothorax. It has some sort of narrowing in places. We don't typically see narrowing in jumping spiders, and it's also shiny and lacks CT, or at least the amount of CT that jumping spiders typically have, and then there's the coloration as well. And as we talked about, they have a lot of audiences to contend with. So how does Synemosyna deal with all of this? So we expect that Synemosyna will have different shape and color profiles when viewed from the top and side perspectives, consistent with the selective pressures at each vantage point. So this is how we tested this. We collected all of these animals. So here we have Colonus sylvanus. This is the jumping spider that is most closely related to Synemosyna that exists in the same microhabitat. So we use that as an outgroup. Then we have the two ant models that we found most commonly co-occurring with Synemosyna, and Periphytopus aurantia down here in the corner is sort of an ambassador for all the other jumping spiders we collected. There were some Hensians, Agobella, Periphytopus, and then in the middle is our star. These names can be a bit hard to differentiate when we're talking about them, since so many of them start with C and have an S sound. So we'll be using some common and shortened names as we move through this. So this is the Sylvana jumping spider, Valentine ants, carpenter ants, sympatric species for the other jumping spiders, and then our star we'll just call Synemosyna. So we'll start for the shape with the shape from the top and side perspective. So this is what we did. Up in the corner here is Anya Viatra. She is an undergrad that helped me immensely with this project, a lot of data collection, animal collection, and data processing. So we took high resolution images and we digitally removed the legs, as the position of the legs can read as different shapes to the R package that we used. And we were just most interested in body shape. We then transformed these into black and white images, and we used the R package Momox that does elliptical Fourier analysis to compare shapes. It then creates an n-dimensional morphological space and places each animal in that n space. And we grouped each set of animals by species, and then we also grouped Synemosyna by age and sex. So to get a picture of the location of these animals in morphospace, we made a two-dimensional version using Pca Analysis. Each point here represents a single animal. I'd like to draw your attention to the cluster of points on the lower half of the graph here. So both the ants are in here. The Valentine ant is the cluster at the top of this red oval, and the carpenter ant, adult male, adult female, and juvenile Synemosyna appear to largely occupy the same space, especially the juvenile Synemosyna there. We also looked at Mahalanobis, if you then look above, you'll see the Sylvana jumping spider and the sympatric species clustered separately. We also looked at Mahalanobis distances. These distances measure the distances between each group of the animals in the actual n-dimensional space. So this can give a really clear picture, much clearer picture of what's happening sometimes. And all of these lines here, the black lines, are the actual relative distances in the n-dimensional space, since we can't really represent that in 2 dimensions to show you guys. But you can see the relative distances. So this is what we found when we looked at the Mahalanobis distances. All Synemosyna are closest to the carpenter ant, and are closer to both ants than any of the other 2 spider groups. So interestingly, they're all farthest from the Sylvana jumping spider, which, remember, is the spider that they're ostensibly most closely related to in their microhabitat. So what we see is that from a dorsal vantage point, Synemosyna has a very ant-like appearance. The lateral shape analysis is a bit more complicated. Everyone seems like they just sort of overlap. However, when we take a look at the Mahalanobis distances, we're able to get a better picture of what's happening here. What we see is that all 3 Synemosyna groups are closest now in lateral morphospace to the Sylvana jumping spider. Adults are farther away from both ants than they are the 2 spider groups. However, while juveniles are still closest to the Sylvana jumping spider, it's still closer to the carpenter and Valentine ants than it is to the sympatric group. So what we're seeing here is a spider-like shape from a lateral vantage point in adults, a spider-like shape from a lateral vantage point in juveniles, with ant-like features as well. So what does all this mean? We have an ant-like appearance from a dorsal vantage point, so this makes sense in the context of airborne or large predators like mud dauber wasps or birds. We have a spider-like shape for a lateral vantage point in adults, and that makes sense in the context of conspecific identification. There could also be constraints here. Female Synemosyna do need to carry eggs in their abdomen, so maybe rather than stretch side to side, they stretch up to preserve their dorsal profile, where most predators are likely to view them from. And then we have this spider and ant-like shape from a lateral vantage point in juveniles. And this makes sense too. They don't have as much need for conspecific identification, but these guys will at some point molt into adulthood. And so there needs to be at some point a transformation from, you know, if they are more ant-like when they're young to get more protection, they do have to at some point become adults. So that makes sense to me in that respect. Okay, so now that we've looked at shape, let's see if color tells the same story. So we took analogous body parts of each of these animals. We collected the reflected spectra from each part, and we averaged it to get a peak reflectance for each body part. And so that's just the color of each body part. We then took these spectra and modeled Synemosyna color in 4 visual models. So we did a tetrachromatic passerine bird and trichromatic sphesid wasp. So these are both major predators of Synemosyna. The sphesid wasps are the mud daubers. So we used a dichromatic jumping spider for both spider predators and conspecifics. And our lab has recently confirmed that Synemosyna is dichromatic. So that was a nice little bonus there. And we use dichromatic ant vision for their putative model. So this table is a little complicated, but it shows how well each animal should be able to differentiate Synemosyna from the two ant groups. Along the top here are the body parts we're comparing. Each set of 2 rows are the visual models. The numbers represent discriminability, or how well the viewer can distinguish Synemosyna from ants. For example, the first 2 rows represent how well the bird model can differentiate between Synemosyna and the carpenter ant and the Valentine ant. Higher numbers indicate that the animal will have an easier time discerning Synemosyna from ants, and lower numbers, the inverse. So for the sake of simplicity, I've created kind of a heat map where the darker grays indicate high discriminability and lighter grays and white are lower. And what we see is that spiders are readily able to tell the difference in color of ants and Synemosyna. Birds and wasps, a little less so. And then ants seem to have the hardest time seeing any color differences. When we look at them laterally, what we see is that, again, spiders can easily differentiate between Synemosyna and ants, but now also birds can as well. We also see quite a bit of discriminability in wasps and more with ants. So when we compare dorsal and lateral discriminability, we see higher degree of discriminability of Synemosyna from ants when viewed laterally, or more of these animals are likely to be able to see color differences between Synemosyna when they're viewed from the side versus the top. So what does this mean? So it seems like they're most discriminable to spiders. That makes sense. They need to be identified by mates, and these are jumping spiders that also have excellent vision. We also have more apparent discriminability when viewed laterally. Again, this makes sense. They need to be identified by mates. And also, a lot of their major predators are aerial. And so there's probably a little bit less pressure to be mimetic. They do have jumping spider predators, but if you're going to be visible to conspecifics, you're probably also going to have to be visible to those. So when we look at shape, we see that Synemosyna is more ant-like from a top-down perspective, where the selective pressures are likely to be primarily predatory, while they have a more spider-like visage when viewed from the side, where they're more likely to encounter conspecifics in addition to predators. When we look at color, we see the same pattern. So taken together, these results support the vantage point hypothesis for imperfect mimicry, that Synemosyna varies in mimetic fidelity based on selective pressures present at each vantage point. And we have support from this from non-mimetic systems that seem to partition their signals based on the viewer. So male bicyclist butterflies have cryptic coloration on one side of their wings and a sexually selected signal on the other side. Schizocosta ocreata shows high contrast laterally against backgrounds that conspecifics see them against, but low contrast from above where aerial predators exist. And our results here, in conjunction with this work in these non-mimetic systems, indicate that considering the location of receivers is important to understanding the evolution of animal signals, and the vantage point hypothesis might apply to more than just mimetic systems. So in the future, I think it would be really interesting to look at animals that are non-mimetic, but are also not as clearly partitioned like these animals here. So something like the mimic, where they appear cryptic all the way around, they may not be. They may have some partitioning that we're not detecting. So I think that might be interesting. So did I answer this question? I think so. I think I answered it a little bit, but we did talk about those other hypotheses, and those also answer the question. And so this is a really fascinating topic to me. And if you found this interesting, I highly recommend this paper here, The Imperfect Mimicry and the Limits of Natural Selection. It goes into a lot of those hypotheses that I feel deserve a little bit better than the breezing over I gave to them. So I highly recommend taking a look at that if you're interested in going into this a little bit more. Lastly, I would like to acknowledge that the land used in this study is the ancestral and present-day territory of the Kaskaskia, Shawnee, and Miami people. The Kaskaskia are no longer extant as a result of the forces of colonialism. So I thank these nations for their stewardship of the land. I mourn the loss of the Kaskaskia people and honor the resilience of the Shawnee and Miami. With that, I would like to thank my wonderful undergraduate helpers, my postdoc who helped me a lot with the mimicry stuff, my committee and my advisor, my funding sources, and my entire lab. And if I have time, I will take questions now. Whew, that was tight. Sorry for being so fast, everyone.
Great, thank you, Alexis. That was so interesting. I really enjoyed that.
Thank you.
All right, chats are moving quickly here.
If I don't have time, I'm happy to answer questions.
I think there's enough time for a couple questions. Do you think that taking movement into consideration would be possible as these spiders walk in a very ant-like manner, especially thinking about the movement of legs that appear like antennae?
So I did look at their locomotion. I only looked at that from a dorsal perspective. I think the problem there is like if you're going to compare the two to see if they have more or less mimetic movement from the top and side, I'm not sure how you would do that, because when you're filming something from the side or looking at it from the side, you just have this linear motion. And as you said, they have this winding trajectory. I did look at their to see if they had locomotory mimicry. I didn't. The results were inconclusive. We used the carpenter ants were really erratic in their motion in some circumstances, and I think that may have. Yeah, I'm still sort of parsing what happened there. But we looked at sinuosity, something called Emax, which is sort of a prediction of predictability of a path, and sinuosity is like how curvy the path is, and we also looked at velocity. So we are looking at that, and they do have apparent locomotory mimicry, and they use their leg one pairs. When they stop, they pick them up and they like kind of wave them around like little ant antenna. So yes, I do think it's important to consider the locomotory mimicry, and I would like to try to find a way to see how that plays out with respect to vantage point, but I haven't come up with something yet. So if anybody has suggestions. Did that answer your question fully?
So, and so that's all the time we have. If you have any more questions, I encourage you to reach out to Alexis either over email or over any of the social interactions we'll have at this meeting.
Absolutely. Thank you so much.
Thank you to all the speakers for your wonderful talks. I really enjoyed them. Thank you to all everyone who attended for the great questions.